Thank you for joining me today. I'm Dr. Steve Pakola, Chief Medical Officer of regenxbio, and I'm glad to share our corporate updates with you today. regenxbio is a leader in gene therapy for more than a decade, with multiple ongoing clinical studies in 2021 across our pipeline of gene therapy programs. Our NAV technology has enabled the development of commercialized therapies and numerous gene therapies in clinical development across a range of therapeutic areas being pursued by regenxbio and our partners. We have a new cGMP production facility coming online, which is expected to allow for production of NAV vectors at scales up to 2,000 liters using our suspension cell culture process. We have an amazing team of experts in gene therapy manufacturing, preclinical and clinical development for the advancement of our internal pipeline programs. The NAV technology platform is a significant advancement over earlier generations of AAV vectors in delivering genes to cells with significant differentiating attributes. These unique benefits have been demonstrated in our clinical studies in animal models, as well as third-party clinical trials in animal models. Our broad portfolio of certain fundamental AAV gene therapy intellectual property includes more than 100 patents and patent applications worldwide, covering our NAV vectors, as well as sequences that are at least 95% identical to NAV capsid sequences. We also have composition of matter claims for AAV7, 8, 9, and RH10, as well as methods for their manufacture and therapeutic use. Our NAV technology platform has been widely adopted, enabling us and our partners to develop best-in-class gene therapy candidates for a wide range of disease targets. This list includes the FDA-approved Zolgensma and several other late-stage programs. We believe that these programs further validate the versatility of NAV vectors and provide additional data that collectively drive the advancement of the AAV gene therapy space. regenxbio is developing a broad and innovative internal pipeline that can deliver gene therapies to a range of tissue targets using monogenic gene replacement and vectorized antibodies. Our AAV-mediated antibody delivery programs use the NAV vectors to deliver genes for the sustained production of therapeutic antibodies in vivo, a novel approach that could be used to provide a one-time treatment for a broad spectrum of chronic diseases. The monogenic gene replacement approach uses NAV vectors to enable sustained production of necessary proteins in tissues. Multiple clinical studies are ongoing in our retinal disease platform and also in our neurodegenerative disease platform, with additional earlier stage programs in neuromuscular and liver indications. We have deep in-house knowledge of AAV production and manufacturing, which provides us with the ability to scale production of our gene therapies while ensuring quality for patients. We maintain a strong network of well-known contract manufacturing organizations, and we are building our own manufacturing facility at our future headquarters in Rockville, Maryland. This facility will provide cGMP production space at bioreactor scales up to 2,000 liters using our platform suspension cell culture process, which will complement our current external manufacturing networks and capabilities. The cGMP facility is expected to be operational starting in the first half of 2022. And now I'll provide a quick overview of our internal pipeline. Our lead program is RGX-314. We believe that RGX-314 has the potential to profoundly impact all aspects of clinical management for patients with wet age-related macular degeneration, or AMD, and diabetic retinopathy, and could be a one-time gene therapy treatment option for a broad range of patients. Current wet AMD treatments are quite burdensome to patients, requiring frequent injections into the eye, which in the real world has proven to be unsustainable, leading to patients continuing to lose vision from this treatable disease. Therefore, a one-time gene therapy treatment option would address a significant unmet need for this blinding eye disease. RGX-314 uses the AAV8 vector to deliver the gene for an anti-VEGF antibody fragment and has been shown to reduce the need for regular eye injections of anti-VEGF. We have been using the tried and true subretinal delivery approach and now also are exploring in the clinic delivery of RGX-314 to the suprachoroidal space. This form of delivery is a non-surgical approach for targeted delivery to the retina that can be administered in an office setting. This slide provides a summary of the current status of the RGX-314 program. We are completing a phase 1/2A study of RGX-314 subretinal delivery, where we have several dose cohorts now out over 3 years from dosing, and later cohorts are out past 1.5 years of dosing. The 3 highest dose cohorts have demonstrated durable and meaningful treatment effect with stable to improved vision following one-time administration. And they have also shown reductions in anti-VEGF treatment burden, which are meaningful endpoints for patients and the clinical management of their disease. We are very encouraged by the durability seen in patients dosed with RGX-314. And based on these encouraging and durable results, as well as successful completion of an end of phase 2 meeting with the FDA late last year, we have initiated a pivotal program for RGX-314 subretinal delivery for the treatment of wet AMD with a planned BLA filing in 2024. Our first pivotal trial, Atmosphere, is active and enrolling patients. We expect to begin our second trial later this year. In addition, we are enrolling patients in a phase 2 trial of RGX-314 delivered suprachoroidally and expect to have our first look at interim data from the first dose cohort in Q3 of this year. This slide shows the stable to improved vision in our higher dose cohorts in the ongoing phase 1/2 trial up to 2 years after dosing with RGX-314. Here you can see that patients in cohort 3 had improved vision over 2 years and an increase of 14 letters. What isn't shown here, but is important, is that these patients are also now out 3 years from dosing and have continued to maintain visual acuity improvement. Patients in our higher dose cohorts 4 and 5, are out now 1.5 years and have shown stable vision, which is still meaningful for these patients. And the reason for that is what I show on this slide, which is that these functional results that we're seeing are in the setting of a dramatically reduced need for anti-VEGF injections. And that is demonstrated in the swimmer's lane plot, in which each line represents individual patients in the highest 3 dose cohorts, with the colored dots representing visits where anti-VEGF injections were given. First, you can see the very high frequency of anti-VEGF injections prior to RGX-314 treatment on the left side, whereas after RGX-314 treatment in the higher dose cohorts, we see clear reduction in treatment injections in cohorts 3 through 5, including many patients who have not had a single injection up to 2 years after treatment. Based on these data, we have initiated pivotal development of RGX-314 subretinal delivery for the treatment of wet AMD. Atmosphere is the first of 2 planned pivotal trials and is active in enrolling patients. This slide shows the basic trial design, which will enroll approximately 300 patients across 2 RGX-314 dose arms versus standard of care ranibizumab monthly intraocular injection. The primary endpoint of this trial is one that has been used historically in wet AMD studies: non-inferiority to ranibizumab based on change from baseline in visual acuity at 1 year. We intend to initiate our second pivotal trial in the second half of 2021, which is expected to be designed similarly to Atmosphere. In addition, we believe we have a clear path to support our cGMP commercial-ready manufacturing plans in the pivotal program. We have initiated the pivotal program using cGMP material produced from our existing manufacturing process, and we plan to incorporate our scalable suspension cell culture manufacturing process to support future commercialization upon completion of a bridging study and the pivotal trials. The bridging study is expected to initiate in the first half of 2021. We also believe that one-time suprachoroidal delivery of RGX-314 can be a treatment option not only for wet AMD, but also for diabetic retinopathy, a larger patient population. We have initiated a phase 2 study to see if RGX-314 can reduce severity of DR and prevent vision-threatening complications such as diabetic macular edema and neovascularization that can lead to blindness. We look forward to the first look at interim data later in 2021. And earlier this year, we unveiled development of RGX-202 for the treatment of DMD, a one-time gene therapy treatment for DMD. DMD is caused by a mutation in the DMD gene, which encodes for dystrophin, a protein involved in muscle cell structure and signaling proteins. Without this protein, muscles degenerate and become weak, eventually leading to loss of movement and independence, required support for breathing, cardiomyopathy, and premature death. Under leadership of our CSO, Olivia Denos, we have constructed a novel advanced microdystrophin, which includes the C-terminal domain. Historically, this has been too large to include in AAV, but we prioritized it as we believe it is critical for cell signaling and membrane integrity. We also prioritized several additional features in RGX-202, including the AAV8 capsid and a muscle-specific promoter, and improvements that may provide higher gene expression, translational efficiency, and reduced immunogenicity. An especially important point is that we have already produced RGX-202 using our commercial scale cGMP process, and will initiate our clinical trials using this material. Proof of concept data from preclinical studies of RGX-202 in the mouse model of DMD demonstrates robust microdystrophin expression in muscle, recruitment of key proteins to the muscle cells, improvements in muscle histology, and meaningful increases in muscle strength and function. We expect to file an IND for this program in mid-2021. We also have several programs in our neurodegenerative disease franchise with one-time gene therapies for the treatments of MPS II, MPS I, and CLN2 disease. We are focused on the CNS manifestations of these diseases using the AAV9 vector to deliver the necessary gene to patient cells in the brain and the rest of the central nervous system. We also use the intracisternal pathway to deliver gene therapy directly to the cerebrospinal fluid. RGX-121 for the treatment of MPS II is currently in a phase 1/2 dose-ranging trial, and we have reported some data from the first several patients, which I will share in a few moments. We are also enrolling patients in a phase 1/2 trial in an ongoing study, RGX-111 for treatment of MPS I, and expect to file an IND for RGX-181. A recent interim update from our ongoing phase 1/2 trial of RGX-121 showed that it was well tolerated in 8 patients across 2 dose levels, and efficacy data so far highlight the potential of RGX-121 to restore biologic enzyme activity and improve outcomes for MPS II patients. Biomarker data from patients in both cohorts indicate encouraging signals of ITS enzyme activity in the CNS, and patients have also demonstrated continued neurocognitive development up to 2 years after RGX-121 administration. Patients have also demonstrated decreased levels of glycosaminoglycans, or GAGs, in the urine, increased ITUS enzyme in plasma, and one patient has shown reduced liver and spleen dimensions several months after receiving RGX-121. This suggests that RGX-121 delivered to the CNS has the potential to deliver benefits to patients outside the CNS. We plan to evaluate a higher dose of RGX-121 in cohort 3 and expect to begin enrolling patients in this cohort in the first quarter of 2021. So in conclusion, based on our NAV technology AAV gene therapy platform, we've made tremendous strides across a range of therapeutic targets in the clinic. This includes a potential first-in-class, best-in-class, one-time gene therapy treatment with RGX-314, which this year entered pivotal clinical development for the treatment of wet AMD. We look forward to presenting numerous additional milestone updates on our programs over the rest of 2021. Thank you.